Hello again and welcome to my beginner's guide to colour genetics in Budgerigars. This is part 5 of the series. If you haven't seen the previous parts of this series, then do go back and take a look at those. So up until now we've considered the gene or the mutation in isolation of the chromosome. We've looked at dominant recessive, we've looked at codominance, and we've also looked at how we can predict the outcome of where we've got more than one gene involved, or more than one mutation involved. But so far we haven't considered the impact of the chromosome that the gene sits on. And that's what we're going to begin to look at in this video when we consider the genes or those mutations that sit on the chromosomes that are collectively or called the sex chromosome. So as I say, we know that the genes are passed on the chromosome, and that will impact how those chromosomes react to each other, will impact on what the potential outcome is. So as I say, in part 5 we're going to consider the sex-linked mutations, those that are linked to the sex chromosomes of our birds. But before we start looking at how we predict the outcome of those particular genes and those particular mutations, we need to understand a little bit about the sex chromosomes in birds as opposed to those in mammals. So that's what we'll look at first. So I suppose we're all fairly familiar with the idea of X and Y chromosomes, and that these chromosomes will dictate whether the offspring will be a male or a female. And in mammals that's particularly true. So in mammals, every mammal will contain a combination of either X or Y chromosomes, and the male chromosome will contain, the male mammal contains one X chromosome and one Y chromosome, and the female has two Y chromosomes. So within mammals it's the X chromosome that dictates whether or not the mammal is going to be a male or not. If it contains an X chromosome, then it will be a male. If they're both Y, then the offspring will be a female. So that's how it works in mammals. In birds it's slightly different, and in birds they don't have X and Y chromosomes. They follow sort of a reptilian or a fish type process in which they have a W and a Z chromosome, so they're slightly different to the mammals. And the birds therefore are also different in terms of which of those chromosomes impacts on whether the offspring is a male or a female. So in birds, a male will have two Z chromosomes, and a female will have one Z and one W chromosome. So within birds, like most reptiles, it's the W chromosome that indicates whether or not the offspring is going to be a female. So the male male will always inherit two Z chromosomes. If it inherits a W chromosome, then the offspring will be female. So that's the way it works in birds and in reptiles, as I say. However, most breeders tend to follow the similar convention, and I'll just talk through that. So we just know that a male can pass on either the X or Y chromosome, and the hen can only pass on the Y chromosome. Therefore the male mammal will determine the sex of the offspring. A cock bird can only pass on the W chromosome, whereas the hen, as I've just explained, can pass on either the Z or the W chromosome. So it's the hen that determines the sex of the offspring. Most breeders, as I say, refer to the bird's chromosomes as X and Y chromosomes, which does start to make it a little bit confusing. However, we will follow that in this guide, and also in the PDF guide that supports this video guide, we will follow this convention as well. So if we look at that in terms of X and Y chromosomes, then the cock bird will have 2 X chromosomes and the hen will have an X and a Y. Exactly the same though, cock bird can only pass on the X chromosome and the hen can pass on either the X or the Y, so it's exactly the same. It's just the reverse of the way we see it in mammals. So for the purpose of this guide, as I say, we will continue to use the convention of referring to the chromosomes as being X and Y chromosomes, just for ease. So what we'll do now is we'll talk a little bit about the Z and W, or X and Y chromosome in birds, and how the two chromosomes differ from each other, and then we'll begin to explore how those differences will impact on the outcome when we start trying to predict the outcome of those varieties or those mutations that sit on the chromosome. So we'll start off by looking at the Z or X chromosome and consider that in terms of one of the mutations that sits on those two chromosomes, and that's the cinnamon mutation. So here we can see it in the cockbird. The cockbird has 2 X chromosomes, and in this diagram here, which is taken from my PDF booklet, you can see that they're identical. So 2 X chromosomes, 2 exactly the same, and it therefore will contain 2 of the genes in the same allele for the cinnamon variety. So this follows the normal convention that we're all familiar with, and that is that, you know, if it contains the cinnamon genes, then we'll look at how the possibilities in terms of the genome and phenotype for that. Now, I'll start by saying that the cinnamon, like all of the other sex-linked varieties, are recessive to the wild type mutation, or the wild type, the original wild type gene. So therefore, if it contains, the possibilities are the genotypes can be wild type cinnamon, wild type cinnamon, and therefore we've got a normal, wild type cinnamon and the mutated cinnamon gene, and therefore we've got a normal split cinnamon, the reverse, so the mutated cinnamon, wild type cinnamon, again, once again, normal split cinnamon, and the mutated cinnamon and the mutated cinnamon, and therefore we have a cinnamon. So it follows exactly the same process as any of the other recessive type varieties, in that you can have a normal split and then the variety, and it must contain both of the mutated genes on the X chromosome for it to be for it to reflect that, for its phenotype to be cinnamon. Notice here where I'm showing that breakdown for the cockbird, that we've placed an X in front of the mutation or the gene. So X cin for X for the cinnamon. The X we've added to represent the chromosome that it sits on, and that's important, as we can see now. If you look here now, here's a picture of the a hen bird that contains 1 X chromosome and 1 Y chromosome. You'll notice that the Y chromosome is completely different to the X chromosome, in that it is actually much smaller, and as far as we're aware, there are no genes for any of the mutations held on the Y chromosome. So there are none of the varieties, the sex-linked varieties, are contained on the Y chromosome. This means that the way the mutations work within the hen are slightly different. So we can see here, we've just put it down, so the hen, which contains 1 X chromosome and 1 Y chromosome, as we've already said, it contain an X chromosome that has the wild type cinnamon gene, and then a Y chromosome that doesn't contain the gene at all, and therefore it will be a normal bird. Or it can contain an X chromosome that has the mutated cinnamon gene, or and the Y chromosome, and therefore it will be a cinnamon. So you can see here that within the hen there are only two possibilities. It either contains an X chromosome with the cinnamon mutation, or it contains an X chromosome without the cinnamon mutation. So again, if it contains a cinnamon mutation, it will be a cinnamon. If it doesn't, it won't. So I put it in red on the slide. The hen cannot be split for cinnamon, unlike the cockbird, which we've seen before. And as I said, there are all of the sex linked mutations are recessive, and none of them, as far as we're aware, sit on the Y chromosome. So therefore the hen cannot be split for any of the sex linked mutations. So that's once you grasp that, it becomes slightly easier. So just remember that the cockbird can be split, the hen cannot be split for the sex-linked mutations. So I suppose one of the best ways of demonstrating what we mean by this is to make a few predictions using the Punnett square. So we've got a Punnett square set up with 3 different pairings here, all representing the cinnamon gene. One of the things you can notice is that we have placed the X in front of the allele that we're focusing on, and it follows the normal process, with the small lowercase being the recessive gene, and in this case the uppercase version, so the large C, represents the wild type. Let's now talk about what each of the birds are likely to be, so what variety they're likely to be, starting at the top one here. Okay, so that there is got 2 X chromosomes, so it's going to be a cockbird, and both of them are the non-mutated gene, so you're going to get 25% of the birds are going to be normal cockbirds. Over here, 2 X chromosomes, so once again it's a cockbird. In this case you've got one of the recessive mutated allele for cinnamon, so it's going to be a normal split cinnamon cockbird. Okay, down here we've got an X chromosome and a Y chromosome, so it's going to be a hen, and this time it contains one of the, on the X chromosome it contains the non-mutated allele for cinnamon, so it's going to be a normal hen. And the final one is a hen because it contains 1 X chromosome and 1 Y chromosome, and this time on the X chromosome the allele for cinnamon is the mutated recessive cinnamon allele, and therefore that's going to be a cinnamon hen. So in this first example, any cinnamons that we produce are going to be hens. Any cinnamons we produce are going to be hens. Of course, of the cockbirds, it would be impossible to tell which of the cockbirds are split for cinnamon and which are just normal, without test breeding them, without testing them in the breeding cage. So you would get, in effect, as a phenotype, you would have 50% normal looking cockbirds, 25% normal hens, and 25% cinnamon hens. You would get no cinnamon cockbirds. Okay, moving on to the next one, once again, where we have a normal hen and a cinnamon cockbird. You can see the top 2 are the same, so those are going to both contain 2 of the X chromosomes and contain one of the mutated cinnamon allele, the recessive cinnamon allele. So those are both going to be normal split cinnamon cocks. And down here we've got one X chromosome and one Y chromosome, so they're both going to be hens, and they both contain the allele on the X chromosome that is for the mutated cinnamon gene. So once again, all of the hens are going to be cinnamon hens. And as for the top one, what we saw here, the very first Punnett square, normal hen with a cinnamon or a normal split cinnamon cockbird, all of the cinnamons produced are going to be hens. Finally, let's have a look at this one. Here we've got a normal and a cinnamon hen, and you can see here all of the hens are going to be normal and all of the cockbirds are going to be normal split for cinnamon. I hope that sort of demonstrates to you how this process works. One of the really interesting things is it would be possible in terms of that, the first two pairings, and if you take the very first pairing, so the normal split cinnamon, if we didn't know that was a split cinnamon, you'd probably expect to get all normal birds. So once we saw a cinnamon in the nest, even before it had feathered up, so once you got those plum coloured eyes of the cinnamon and the whitish down, we would know that they were going to be hens, and we would know that the cockbird was split for cinnamon. So it's possible very early on to know which of the birds are going to be hens when we've got when we spare a normal hen to a cinnamon or a normal split cinnamon cockbird. Okay, so that's the how the recessive allele works on the sex chromosome, and this would work for exactly the same process would work for any of the sex-linked allele or sex-linked varieties, such as the Ino or opaline, for example. So they would work exactly the same as this.